Hi, good morning. Welcome back from the vacation. And we continue with systems biology. Let's take a nice deep sigh of relief. In the course feedback, thank you for all the feedback. We looked at it, there wasn't some overwhelming thing that wasn't balanced by the opposite overwhelming thing. So I try to especially give some more biological examples in detail. This I learned from the feedback. Some people mentioned that to drop the deep breathing, but I'm not going to do that. I'll just say that you have your autonomy. You don't have to take a nice deep sigh of relief when I suggest it, but if you do, practice it. Most likely you'll enjoy it and get into the relaxed state that I'm aiming for. All right. So in the last lecture, we talked about bacterial chemotaxis. So in this lecture, we'll talk about fold change detection. That's the title. So I want to make a small review. Let's start with where we are on the course plan. So this is the course. We finished one part of it, which had to do with the principle of network motifs. That basically complex networks in biology are built of a small set of circuit elements that we can analyze mathematically, see what their functions might be, then test it experimentally. Based on some questions and exercises, I just want to stress, if you have a network motif like this, it's almost always connected to some other things. Right? So the question is, can it have some autonomous function despite the fact that it's inside a big network? And the experiments so far on the best studied systems say that yes. The functions like speed up, stabilizing the steady state, sign-sensitive delay, temporal orders and stuff like this can work even if this small circuit is embedded within other interactions. That was the first part. And now we're in the second part. We're discussing the principle of robustness. This principle says that circuits, biological circuits, are designed or evolved to work despite the natural variations in inputs, parameters. We talked a lot about protein concentrations. So even though the circuit is made of proteins, it might need to make a very precise computation, like understand exactly how much osmotic pressure is outside, and it translate that into the amount of phosphorylated transcription factor inside precisely, and how you could do that even though the proteins in the system can vary by, let's say, twofold between bacterium and bacterium, even if they're genetically identical. And we saw how bifunctional proteins can do that, paradoxical components. We talked about robustness in bacterial chemotaxis that's carried out by integral feedback, exact adaptation, talked about that. And we're continuing, we're still in this part, and we're going to talk about additional principles on how you can work despite huge variations in background signal. I'll tell you more about that. So that's our orientation, where we are. I'm going to erase this now, okay? And as a review about what we did last time, we talked about bacterial chemotaxis. Remember bacteria swimming, running, tumbling? Controlling their tumbling frequency. Why? because maybe they want to get to a source of attractant. So here's a pipette with some attractant in it, creates a gradient. The bacteria know how to climb this gradient. The way they do that is if they're going in the right direction, is if attractant concentration is increasing, they reduce their tumbling frequency, so they tend to keep going in that direction. And if it's decreasing, they increase their tumbling frequency. So statistically, they have more chance to go out of the wrong direction. And this way, slowly the cloud of bacteria moves up to the mouth of the pipette. And they can do that over at least 5 orders of magnitudes of concentrations. So big concentration range. And if you look at the computation that they're doing, this tumbling frequency, you know, remember it comes from a 6-protein circuit that has phosphorylation and methylation, adaptation, etc. So this tumbling frequency output is about 1 per second. And if at this point I put in a step of attractant uniformly in space, the bacteria stop tumbling for a while and then they adapt. So they have exact adaptation. And what they're actually doing is sensing the change, like a derivative, you can say in a certain way. And it's important for you to remember this concept of exact adaptation. The steady state level doesn't depend on the steady state amount of input. So if I put another step here, it will happen again. Steady state output doesn't depend on steady state input, exact adaptation. And the second thing I'd like you to know, this is going to be important for our course, is how this happens. And the idea is that the receptors in bacterial chemotaxis are methylated. They can be methylated on several sites. And the rate of methylation is controlled by 2 proteins, R and B. And the biochemistry works that this B only affects the receptors which are in their active state. And these active receptors make the tumbles. And this means that the only way you can get steady state is if this activity, which goes like the tumbling frequency, so I can write here A, this time, goes to A0. That is to say, and this is A0. So built into this equation is that the only way to get steady state is if A goes to this A0. It's like the thermometer I talked to you about last time. When you have temperature control and you have the power to the heater depends on the change of the power to the heater depends on the difference between the temperature and the desired temperature. So this gives robust, exact adaptation. Power, temperature, set point, T0. So this integral feedback is the way that engineers and also biological systems use to get exact adaptation. They integrate the error between the activity and the set point activity. How do they integrate it? By changing this methylation level. So if I plot here now the methylation on this graph, when I add attractant, the methylation increases. And when I add more attractant, this step should be here, right? When I add more attractant, it increases again. That's the internal memory of the external signal. But because methylation, it balances out the effect of attractant on the receptor, this kind of feedback makes sure that methylation increases exactly and stops increasing when activity reaches its set point. Because methylation receptor stops changing only when the activity reaches its set point. And this set point is given by the ratio between these biochemical parameters of Kr and Kb. If I change the amount of Kr, change the amount of Kb in the cell, the set point changes. but still has exact adaptation to the same different set point. Let's take a nice deep sigh of relief. Welcome. Any questions? Yeah, it's been a long time since. Yeah.
The attractant, you say it's in the gradient form. So if it's not in a step, if it's not in a step function form, how would the M vary?
Good. So the question is, in real life, bacteria are living in a gradient, in a continuous spatial gradient of attractant. In the experiment, we're doing something quite artificial, mixing it quickly with a lot of attractant uniformly in space in order to get a good something good for theory, I would say. It's like a nice good for theory. If you think about the bacteria moving up the gradient, what it sees, the signal that it sees in gradient, the bacteria is doing this. So what it sees, attractant versus time. If it's going up the gradient, attractant increases and it decreases. See something like this. So with the same circuit, it's always trying to respond and adapt, and you get quite a complicated output. But you still, with these equations, you can compute it. And you know, if you guys are so inclined, you can do a computer simulation where you take agents like bacteria that move and change direction randomly, and the rate of change of direction is governed by this equation. And you can see on the computer how this cloud of agents walks up the gradient. You can also linearize, do linear theory here, and solve a lot of things, even the flux of the cloud. But I don't want to get into that. Did I answer your question? Maybe not. So,
Kind of.
what?
I remember seeing somewhere that the correlations with the second derivative of the
Concentration.
I took the course. I think Michael Eisenbach,
Yeah.
and I recall something about there being correlation with second derivative of the attractant.
Yeah. So what you're saying is that there's something about the second derivative of the attractant, if it looks like this or like this. So this is a maximum, this is a minimum.
Yeah.
And then you can linearize, but that's sort of too technical for this course. And I want to say there's quite a lot of theoretical papers on bacterial chemotaxis, and if you're interested, I can even, if you're interested, you can tell or email the course Mitergelim, and we'll put some on the website. If you're interested, because you can do beautiful theory on this system. All right. I'm going to erase now, and we go to fold change detection. But you have to remember, exact adaptation and integral feedback will play a role. Bacterial chemotaxis, all of this. So even though I'm erasing it, keep it in close to your heart. All right. Okay, so our topic today is called fold change detection. Imagine that you're holding a kilo in your hand. You get used to it. And then I come in and I start putting in, you're not looking at your hand, I start putting in little weights on your hand and ask you, when can you first detect? And you suppose the answer is 10 grams, let's imagine. Okay. Now we do it again, and I give you 2 kilos. And I start putting weights. It turns out that your detection threshold will be 20 grams. And if I put in 10 kilos, it will be 100 grams. What connects all this? It's like a ratio. The minimal detectable difference is proportional to the signal that you've adapted to. Okay? This was discovered by Weber in the 1850s. This is Weber's law. And we detect relative changes. So it's easier to detect the difference between 1 and 2 kilos than between 10 and 11 kilos. Same thing with vision. You go into a dark room with the one candle, and you adapt to the light. It takes you a while, but you can adapt, right? Even if you were in sunshine, bright sunshine, you go into a dark room, after a while you can see contrast differences. Now I bring another candle, things get much brighter. But if there was 50 candles, and I bring in the same one candle, same absolute difference, you can barely notice it. The visual system also works by ratios. Even though it's one candle difference, the absolute difference is the same. The ratio is, it works on ratios. Okay. More precisely, I want to say that most of our senses need to remove, cancel out, normalized by a background that multiplies signal. For example, let's stick with vision. Suppose I want to look for something, like I lost my iPhone, I want to find my iPhone here in the room. And thank you for giving me your music and your iPhone. What I'm seeing is the contrast is a function of position, which is, what color each pixel is black, white, gray. But this has to be multiplied by the ambient light. How many photons come from the lamp or the sun or something like that. And that gives me my input to the eye. So what I care about actually is the contrast, because I want to find my iPhone. But I have to work in quite a wide range of ambient light. For example, the difference between starlight and full sunlight is 10 orders of magnitude in terms of photon flux. But our eyes work on quite a large dynamic range, and the search they do, the time it takes me to find the iPhone, is for a large range of backgrounds, is the same. So eyes can search for an object with the same timing, you can say timing or efficiency, over several decades, orders of magnitude of eye. So in other words, our visual system knows how to divide the eye, get rid of the eye. Not the eye, the eye, the intensity, the background intensity. Chemotaxis. If we put our pipette here with some S0, the source S0, and I ask you what the gradient is of attractant, if it was just, what I'm going to say is that the attractant field is also multiplied by something. That something now is going to be the source strength, this source strength. Those of you who know about diffusion will know that what you have from, if you go from a point source, you have some function where things kind of start diffusing out, but it's linear with the source strength. In other words, if I double the amount of attractant in the pipette, all the gradient will double at every position and every time point. So again, if the bacteria want to go into the pipette, that's what they care about. And indeed, they can do that, as I said, across 5 orders of magnitude of concentrations. And they do that with the same speed, the same efficiency over 5 orders of magnitude. That's to say, I take this pipette of some attractant, and I put it in a little cover slip that has bacteria swimming. And I count after 1 hour how many bacteria went in. Okay? So I take out the pipette, I wash it. Then I take the contents and put it on a Petri dish and count colonies. And over 5 orders of magnitude, the number of bacteria swimming in is the same to within a factor of 2. So they know how to, their speed is that they find it is insensitive to this multiplying the entire field by a number, S0, that can range between over 5 orders of magnitude. Okay, so that's, again, two different sensory systems. They have to deal with multiplying the input field by a number that you have no control over. No? So we already saw ambient light. We have attractant source. And here's something, I'm going to say something now that touches, again, very close to the heart of the cell biologists here and molecular biologists here, especially those working on animal cells like mice and human, flies, worms, and all the other pets the biologists love, zebrafish. There's another situation like this. Here I'm talking about the senses of an individual cell. So this is what an animal cell looks like. It has a nucleus. And inside the nucleus is all the DNA and all the genes. So if you want a transcription factor to turn on genes in response to a signal, it has to go into the nucleus. A lot of times the transcription factors are outside and they go inside. Am I right, biologists? Of course I'm right. Except when I'm wrong. So it's just like in bacterial chemotaxis. There's a receptor that changes the senses the signal. And the thing in animal cells is more complicated than bacteria. There's usually a cascade of events, phosphorylation, 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 for some reason. It's weird, but... And in the end, a transcription factor, so I'm going to be, I'm actually going to name it based on your feedback, okay? I'm going to name the transcription factor. This transcription factor, NF kappa B. What does it do? Who knows NF kappa B? Who's heard of it? Just raise your hand, be proud. Come on. Biologists, pride, pride. I'm a biologist and I'm proud. So what does it do? Who can tell me? Don't be shy. He asked. He opened his hands. Huh? Yeah, part of the immune response, right? So like, I think, if I'm not mistaken, if there's a viral attack, it can cause inflammation. So that's downstream genes here have to do with molecules that call the immune system over, right? Something like this. What else? Huh? Cell death. In what situation? It calls cell death when the cell needs to die, right? It's a good death. I'm not saying any of you should commit suicide. In fact, no blues musician who's only played the blues ever committed suicide. Eran Sabag told me that. But cells can commit suicide. It's good that they commit suicide because if they don't, they can, let's say, turn into cancer cells. So one of the first things cancer cells do is turn off their ability to commit suicide, for example, so in certain ways. So this protein is important, and when it's deregulated, it's not working right, it can lead to diseases, all kind of nasty diseases. This NF kappa B. So you'd think it's really very precisely regulated, right? Okay, so let's say there's a signal coming in. It could be a stress signal, viral signal, or a chemical called tumor necrosis factor. Necrosis means death. Tumor means cancer. Factor means... usually it means in biology something we don't understand yet. So a lot of things are called factor, and then you discover it's a protein. So, am I right? So, like factor X, and okay, tumor. But then it's of course well known nowadays what this molecule is. So suppose we give here again a step over time, remembering that, like you said, in the real world it's gradual, perhaps, but maybe there's some viral attack and it's really sudden. It doesn't matter in some way. And now I look at how much NF kappa B is in the nucleus. So for instance, if I were able to stain this protein and count how much of it in the nucleus. Why the nucleus? Because that's where the DNA is, that's where the genes, that's where it can act to activate genes. And I look now with this new technology. Miri, did you get an answer to my question? What's the answer? Yeah. Yeah, so this is brand new data, okay, from Suzanne Gaudet in Molecular Cell, 2014. Okay, March 2014. So right off the press. I was really excited when I saw this paper. So thank you, Suzanne, if you're watching this. They were able to look in individual cells, and first thing they see is without any signal, there's already NF kappa B, and each cell has a different level. That's already something to worry about, because and then when you add the signal, basically each cell responds. And a lot of these systems, they also have exact adaptation. That's not important right now. And she asked, what's the most constant between cells? So if everything's changing between cells, what's the most constant? So if this is the level Z1 and Z2 in a certain cell, it's Z2 over Z1, which is the fold change, is very constant between cells. Though the absolute levels vary by, let's say, fourfold between cell and cell. Okay, so then she said, okay, so we need, in order for the cells to respond, it could be that after this signal, they only increase, let's say, by 30%. Right? But there's a fourfold difference between cells. But each cell over its own background increases by 30%. So in order to actually work, this system needs to adapt or to factor out its basal level. So there's another, in this case, it's the basal nuclear level of transcription factor that needs to be normalized out. And you need to only worry about the ratio, because that carries the information. So I hope by now I've convinced you to death that biology needs a way to sense relative changes. And Suzanne Gaudet and her team went on to show what circuit does this here, downstream of NF kappa B. What circuit, what network motif does it? And following up on the work that we did to start up this question in 2009. So 5 years. 5 years this concept, let's say, is available. So last time, when I wrote the book for the first edition, we didn't know about this yet. But now we get a sense that it's so crucial for systems biology. So it's quite satisfying to. There's a lot more to discover, and this is my message to you. Yeah.
Does there have to be a motif that makes the fold change essential and not some adaptation of the cell that it needs higher levels in order to respond? Maybe the cell is less sensitive to begin with, and that's why it has higher basal levels and needs higher levels to respond to?
So the question is, I understand correctly, maybe there's a difference between these cells. These cells are less sensitive, or these cells are less sensitive, so they need higher signal. I agree, and the question is, what does it mean that they're less sensitive? And we're going to find out.
Maybe general machinery of some sort.
It could be, in principle, it could be general machinery. The problem is if you look at other nuclear proteins, so there's many other nuclear, the ones that's high in one isn't necessarily high in the other.
So it has to be pathway specific.
It has to be pathway specific, gene specific, And but instead, let's keep that. That's also a possible answer. It's something global. Maybe that's also a case. We don't know yet. Right? So it's just the beginning of this field. Yeah. All right. So I'm going to erase. Can I erase now? We can relax now. We did so much. Weight, vision, chemotaxis, nuclear entry of NF kappa B. And now let's talk about this property that's called fold change detection, make it more precise. Output. Signal S of t is exactly the same as to the signal P times S of t, where P is a constant. In other words, if I give an input signal that looks like this, and I multiply it by P, the output will be and you might worry about initial conditions. So this is exactly when you start from adapted initial conditions, steady state. If you start from different initial conditions for the two, you exponentially converge to exactly the same trajectory. That's guaranteed. So that's fold change detection. But let's do it a little bit more simple. So if I give 2 steps of input, so this is time, and I go from level 1 to 2, and then I go from level 2 to 4. What's special about these 2 steps is they have the same fold change. What's different about those 2 steps? The absolute difference is different. Okay? The output of the system same output curve. So if I take these 2 curves, I plot them like when I was a student, we had transparencies. It's no longer now. We used a viewgraph. We projected on a wall. If we took these 2 transparencies and put them together like this, you'd get the same curve. Same amplitude. Same adaptation time. Same everything. Same steady state. That's the property of fold change detection. It's a tough property, those of you who know dynamical systems. Not many dynamical systems can do this. So it's a big constraint. Notice that at level 2 and level 4, the steady state is the same. Why is the same? Because all the curves are the same. So, of course, the steady state is the same. Also at level 1, it's the same. So this means that fold change detection means exact adaptation. So every system with fold change detection has exact adaptation, but it's not the other way around. Because if I had a curve like this, goes to the same steady state, that has exact adaptation, but it's not fold change detection because the 2 curves are not superimposable. So fold change detection. Full change detection means Weber's law that I mentioned early, but not the other way around, because Weber's law only talks about the maximum amplitude and not about the total shape of the curve. So it's kind of, this concept has in it the features we know from sensory systems for 150 years. Exact adaptation, like our visual system, our nose, our ears. Weber's law, a kind of a generalized Weber's law. But there's more. There's also all the time scales, etc. And that's what looks like is going on in cells, in biological cells, this fold change detection. So that's what we're going to talk about, fold change detection. And it's hard for a circuit to make fold change detection. So just, for instance, if we remember simple regulation, does not have fold change detection. Let's analyze why. Our input signal here makes X active. And what happens to Z? It rises up, right? Like this. You know this exponential rise, etc. This doesn't have fold change detection, of course. Of course, first of all, you can see it has no exact adaptation. It can't have fold change detection. It just goes up. If I make another step here, it'll go up. If I take this curve and put it here, it would look like this. The response is dependent on absolute signal level, not on relative changes. Not only on relative changes. Here, the response is only determined by the relative changes. So simple regulation doesn't have fold change detection. None of the network motifs we talked about has fold change detection, except for one. Okay, so you can rule out basically the vast majority of the circuits that occur in biology or that don't occur in biology, but only one can. I'm going to tell you about it. So is it okay to erase already? Kid, there's a question.
Can't you say that there is? The question is, why do you have to have exact rotation or have fold change detection? You could have the same graph but shifted upwards, and it would still respond to the fold change, but it won't return to the same level.
For fold change detection means that you have to have, suppose you have something like this for this step, and then for this step you have something like this, and I want this graph to be exactly like this graph. The only way to do that is if both graphs have the same steady state level, because that's part of the graph. That means the steady state level doesn't depend on the input level, on the steady state input level. And that means exact adaptation. So I'm not talking about being able to shift the graphs up and down, and I mean exactly the same response. Okay. Okay. So the incoherent feed-forward loop can show fold change detection. So for fold change detection, you need to remember the background level when you make a change. That means you need an internal memory. That's why when so the origin of this was when Mark Kirshner and we, in our lab, we made two discoveries. We didn't publish them. We didn't understand them. And when I went on sabbatical, we understood that both of them mean fold change detection. The one is in the Wnt signaling system. Determines developmental shape. And the other one in the ERK system, the MAP kinase nuclear transcription factor. It's not important right now. Around 2008, something like that. And Leo Gontoro was a postdoc. Now she's a faculty member in Caltech. Mark Kirshner. And And we thought, what kind of circuit can have a memory, and that also occurs often in biology, and that can give you adaptation? So incoherent feed-forward loop. Okay, so incoherent feed-forward loop, as you know, looks like this. This is the type 1 incoherent feed-forward loop. So it must be a relief to see back, a nice friend like an incoherent feed-forward loop that we saw a few lectures back. So let's welcome it with a nice deep sigh of relief. Say good. There's some regularity in the world, in this chaotic world. And in order to see that it has fold change detection, you need some kind of division to happen there, something dividing something out, right? So we need to write a more precise model of the incoherent feed-forward loop than we did before, where we used logic step functions. So I want to talk a little bit about how we can model the repressor. And here I'm going to do some math that also explains all this Michaelis-Menten equation that we're talking about in this course a lot of times. So the answer will be, okay, that the production rate of z is beta divided by 1 plus y over k, where this is the dissociation constant. This is the answer. Okay? But how do you get that? You think of the DNA, and this is y, and and this is the site on the DNA where y binds in front of gene z, and its purpose is to repress it, right? And it binds the site with a rate k on, and goes off the site with a rate k off. These are the two chemical rates that have to do with how fast it finds it, and when it finds it, how fast it falls off. They have different units, as you'll see in a second. So this happens quickly, let's say on the order of once a second. But since transcription, translation events, as you know, happen on this time scale of hours or minutes of hours, we're going to average over many, many binding, unbinding events and ask, what is the probability that the DNA site is unbound? When it's unbound, then there's transcription at a rate beta, because this is a repressor. When it's bound, there's no transcription. When it's unbound, there's transcription. So that is bound 50% of the time, 70% of the time, 100% of the time, 1% of the time. And the way to do that is to notice that we have the DNA, and the DNA time bound to y. This is the total concentration of the site in the cell. Maybe 1 copy per cell, doesn't matter. Okay? So this DNA can either be free or bound. And the rate of change of this let's say the rate of change of the bound state is if the free state meets a y, and this is the rate. So this is y meeting the free DNA site. And this is the bound site dissociating. So it's dy k on minus d total minus d k off. Because d plus, I move d times y here. So this is like d. And at steady state, this equals 0. So steady state, because I'm averaging over time, is much larger than 1 second. And that means that d y k on plus k off equals dt k off, or this open state is dt over 1 plus y over k, where k equals k off divided by k on. How did I do that? How did I do that magic? D over dt equals k off divided by y k on plus k off. And I can divide by k on, by k off. Yeah, I divide by k off, so it's 1 over y divided by k plus 1. And k is k off divided by k on. Okay. So I'm going to, you can write, this is the fraction of free site. Fraction of free site. And the final answer is the transcription rate. Transcription rate of gene Z is beta times this fraction. Great. Any questions about this? It's going to be important. I dwell on this because from natural statistical mechanics of basically a ball and a site, and the ball binds and unbinds, and this free energy of binding, all these k's, this k is e to the minus delta of g, g is the free energy. You get something that looks like fermions, basically. This site can be bound or unbound. And this particular form has y down here, and that's going to allow us to divide things, basically. So that's where y is the memory. When I have x, z gets turned on, y remembers x and turns z off. That's the principle. But the way it turns it off is proportional to its concentration. Hey. I'm going to erase it, okay? Some of you, it's easy, this computation. Other people are in shock. Like 100 years ago in the trenches, World War I, you've just been hit by a big, big Turkish shell. In, what was that battle? The big battle, the Turkish versus the, Gallipoli, yeah. Do you know this? 100 years ago, and 99 years ago. All right. It's okay. We're not in the trenches. It's not World War I. Even though each of us has its own trench in our life. And it's World War I in our life also. It's not stopped. Okay, so when we put in the signal, y is made. Z first goes up, and then because y is a repressor, goes down. So you can see this has the basic elements of fold change detection. But fold change detection is quantitative. We want to make sure that if we do another step now, same fold change, we get the same response, right? So we want to see if we can do that. So we need to write down the equations here. The rate of production of y is basically, x star minus alpha y, and this means that y can rise. And what about z? So far we talked about the binding of y to its site in the Z promoter. This is the repressor y. But X is the activator, also can bind there, right? And it also has its binding and unbinding. And when you work out the same kind of math, and you ask yourself, when is Z transcribed? Because X is an activator and Y is the repressor. Okay, and let's assume that Z is transcribed when X is bound, but not Y, right? Because X is the activator and Y is the repressor. And we ask, what fraction of the time, averaging over this rapid binding unbinding, is the site in this state, X bound and Y not bound? We do the same kind of calculation. We get to the conclusion that the transcription rate of Z is beta X divided by 1 plus X over K1 plus Y over K2 plus XY over K3. And here we're counting all the possible binding states. This is just X binding. This is just Y binding. This is both of them binding. It's also possible. And each one has their own dissociation constant or halfway point. But I want fold change detection. And it's not going to work with this complicated, messy thing. So I'm going to take an important limit. Okay, so the important limit for FCD is that Y, the amount of Y transcription factor, is much larger than its K X, yeah.
Numerator of K3 in the
Here?
denominator. Yeah. So over it is it XY or X4?
X times Y. Thank you. Thank you. Really excellent. These kind of questions I want. So if you don't understand something, please ask. What else? Why is it X times Y? Because this is the probability that both X is bound and Y is bound. So they need the product of both concentrations. It's like they're there at the same time. And you can derive that with the same kind of algebra that I just did by asking yourself all the possible states, and just like I just did with the case of just a single Y. So I'm going to assume that there's a lot of Y, basically. The Y is almost saturating the receptor. And that X is in its linear regime. And then what we can write is X star over Y times the number minus alpha Z.
Where is the assumption that X is in linear regime? Instead of writing something after Y.
Yeah, it's here. It's here because here, well, it's not only here. It's also that X is much smaller than K1. So I can neglect X here. I have to keep it here. Neglect it here. And neglect, basically I need to neglect everything, the 1, the X, and the X times Y, and keep only the Y. So Y has to be the biggest thing here. Y has to be high level of repressor. The repressor needs to be almost always shut off. But it's sometimes open, and on that sometimes open you can play and get fold change detection. By the way, some promoters are built so that X and Y bind the same site. So the activator binds the site and repressor binds the same site. And then this term equals 0, or K3 equals infinity or something like that. And then it's even easier to get fold change detection. So that's a competitive situation is even easier to get fold change detection. So not every incoherent type 1 feed-forward loop will give you fold change detection. It only happens at a certain range of parameters. So that's important to know.
Explain again, please, the production term of Z.
This?
No, the Z equals.
This?
Yeah.
This is the transcription rate that comes if so in my limit, I can neglect these because this is so large. There's another K2, right, that goes here. So let's write it here, right? Or you can put it into the same parameter, like swallow it inside the beta.
You can neglect the third term because Y is very large. So unless K3 is much larger.
How can I neglect this?
Yeah, how do you neglect it?
So X is very small.
Yeah.
So the point is that the probability of X binding and Y binding is smaller than just the probability of Y binding.
Oh, it's much smaller.
Yeah. So that's why. Or competitive binding, that is X and Y bind the same site, and then this term is then K3 is infinite, basically. This term is 0 if there's competitive binding. Competitive, because X and Y cannot bind the same site. So then I don't even have to worry about this term. That's really good questions. I hope you're writing them down. You're writing them down, yeah.
So the K's are of the same order of magnitude?
They don't have to be. The K's here are the same order of magnitude. They typically they scale with the well, they can be over a wide range. It has to do with the, let's say, the chemical structure here, how quickly X falls off the site. So it could be a millisecond or a tenth of a millisecond or 10 milliseconds, and that means you can have, let's say, 2-3 order of magnitude change in K. So you can tune it. And that's why you can keep, if you have 1,000 transcription factors in the cell, K can be equivalent to 10,000 transcription factors in the cell, and then X over K is a small number. Or it could be a large number, and that's tunable by evolution, you can say.
So if you didn't have any assumption regarding the last K, the K3, so how you don't have a scale?
Okay, so okay, maybe I'll elaborate a little bit on K3. Yeah. So I'll teach you a little bit more biology of promoters. Okay, just you can try to follow me if you can. So there's something called independent binding, and that's under assumption that X and Y can bind simultaneously. Then the binding, this is the probability that X is bound, and this is the probability that Y is not bound, like I showed you, and you just multiply them because it's independent binding. And then what you get is X, okay, over 1 plus X over K plus Y over K, K1, K2, plus xy over k1 times k2. And in this case, k3 is just the product of the two k's. That's fine. That's called independent binding. But you can also have exclusive binding, which is if evolution, in its infinite wisdom, made them bind the same site. Okay? And then the answer turns out to be this. It's not possible for both of them to bind. And in the case of NF kappa B that I described, it's actually this exclusive binding that's working there. So there's a lot of details you can play with the design of promoters. And okay. So evolution has to work, you can say, to get the k's right in order for this equation to function, but it's certainly feasible to do it. Okay, so it's not any incurrent feedforward loop, but some incurrent feedforward loop. And then this system of equations, has fold change detection. It has fold change detection. It has fold change detection. It has fold change detection, and you can prove it. How do you prove things like that, or show things? Of course, you can simulate it on the computer, and I think you'll get a chance in the exercises to simulate it on the computer, and try to see if the two curves overlap, and play with it a little bit. And if this assumption is almost right, if there's a little something plus here, then it's almost FCD, fold change detection. So it's like this graphs almost overlap. So assumptions like this are only right on paper, but in reality, there's always some contribution from these other terms, and then fold change detection is, let's say, 1% off, or it depends on the correction, but it's still within experimental error. Looks like fold change detection. So basically, Y is changing continuously, but it's compensating exactly for the fold change. Let's for instance look at this. Let's look at the steady state, because we want to see if it has exact adaptation. So exact adaptation means the steady state shouldn't depend on this input signal. That's exact adaptation, right? So does that happen here? You know what? Maybe let's do this as a little exercise right here in the class for you guys. So try to pull out a piece of paper and calculate the steady state here. The steady state of Z. Steady state of Z. Please try to calculate it, and you can also turn to your person sitting next to you and see if you can. Let's see what the formula is. Some of you aren't writing. I'm puzzled. Why that is. Thinking by your head. You want to do it in your head? No, do it on paper. Then I don't want to burn your brain out. It could be different or same, doesn't matter. I mean, the answer changes, but it's still the exact adaptation. Does anybody want to read out the answer? Don't be afraid.
Y steady state beta K2 over 8. I mean, y is the bottom of the steady state, y steady state. And your z steady state.
Right.
No, the purple.
Here?
Yes.
Yeah.
It should be y instead of y steady state.
Right. So when we look at the steady state of equation like this, we in steady state both equations have to be equal. Steady state is when all changes are 0, right? So from this equation, you see the y steady state is x star divided by alpha. So that's nice. Y is proportional to the signal. It remembers the signal. We want that. And then z steady state, this equals 0. So z steady state is this divided by alpha. And we can plug in what y steady state is. So it's x star beta k2 divided by alpha, x star over alpha. By the way, if there was alpha 1, alpha 2, here would be alpha 1, alpha 2. Doesn't matter. And you get something. The remarkable thing is it doesn't depend on the input signal. Okay, so there's exact adaptation. But it's more than exact adaptation. The trajectory tracks, if I change x star by any factor, the trajectory will be exactly the same because y normalizes it out. And you can again prove that by dimensional analysis, by going to dimensionless variables. But we'll get to do that. So simple regulation has no fault change detection. Incoherent feed-forward loop has fault change detection. And I want to connect this now to this NF kappa B system I started with, Suzanne Gaudet's experiment. Because she kind of went through to ask what the circuit is that downstream of NF kappa B. And it turns out it's an incoherent type 1 feed-forward loop. So NF kappa B, turns out, works as a dimer by binding another protein, And that's the active transcription factor. But NF-kappa B also activates the gene for that protein. And that protein can also dimerize with itself. And when it dimerizes with itself, it binds to the same site as NF-kappa B, but doesn't cause transcription. See what I mean? So there's this detail in the NF-kappa B system that there's this homolog or protein that looks like NF-kappa B, but doesn't cause transcription. And she could show that if you, using genetic tools, perturb the level of this guy, you lose the property of fold change detection in the system. So I'm guessing what I'm saying is that this network motif, the incoherent feed-forward loop, which for about a decade we were thinking has certain functions like speeding up and making pulses, might be a very useful circuit in animal cells to get rid of some built-in imprecision or variation in the machinery, which has to do with basal levels of transcription factors. And I maybe just mentioned in the end that you can ask, is this the only kind of circuit that has fold change detection, or are there other possibilities? And you can prove a theorem that limits basically, or sufficient conditions, sometimes necessary and sufficient conditions for fold change detection. And you can find basically another class of circuits that have to do with integral feedback loops. So you have these kind of systems. This has integral feedback loop because the steady state is always Z0. That's like what we talked about before. That's the only way you get steady state here. But it has no fold change detection because this is a linear system. And if I change the input by a factor of 2, the output will change by a factor of 2. It's a linear system. It can't have fold change detection. But using this theorem, you can repair integral feedback to make it have fold change detection, basically by sticking here a y here and making this proportional. This nonlinear integral feedback. If I draw this, the signal activates Z. Z increases Y, but Y decreases Z. So it's kind of a negative feedback loop. But Y also autoregulates itself because of this Y term here. This this particular circuit is found in bacterial chemotaxis. And underlies this ability to make the search invariant to 5 decades of attractant. All right. Ken.
So what do you need Y to be to get
What do you,
It's one, so you don't get it, but otherwise,
if you, can you repeat the question?
what do you need y to be? So you said if it's a linear system, then you don't get full change.
Yeah, if it's a linear system. Why don't you get full change detection? Or
You said that if it stays as a linear system,
right, the linear system. It's the input and something like that, right? The linear system.
yeah. And then you said, well, if you had just
Yeah.
the additional factor is using
The y here.
y, yeah.
Yeah. Multiply by.
Yeah, but it has to have some kind of range because well, you need the part to be, I don't know, scalar, you need it to be square roots. I mean, what will give you the
Here, the y here.
yeah.
Yeah, it has to be just y times k times y times. Sorry, I didn't write this. Just multiplying by y here. What does this does, you can say, is it makes the speed of returning to the steady state bigger the bigger y is, or something like that. But the reason for all this is that this is a nonlinear system because you have y times z. So in that sense, it's not linear. And you also have x divided by y, so it's a nonlinear system. The reason is that if I multiply x by a number and y by a number, the same number, then here there's no, z isn't affected, but y also you can erase, because there's a y here, you can erase this p. So all the equations remain invariant. So basically that's what you need to do, is you need to ask, if I increase the input by a number and the memory by the same number, what kind of equations are invariant to that? So if I increase y by p, I have to put a p here, and a p here, and a p here, and a p here, and it just cancels out. Then the linear feedback, if I do the same thing, I have to put a p here, and it doesn't cancel out. And I have to put a p here, and a p here, and it doesn't cancel out. So that's the heart of the theorem. So you look for equations that have this homogeneity condition, it's called, where if you do this transformation, equations remain invariant. And what time is it now?
How is the first equation homogeneous?
It's homogeneous. It's not. It's okay. I'm going to write down the theorem because we have just a few more minutes, and then you'll understand everything. So this is called the Chauval et al. PNAS 2010. Sufficient conditions for FCD. So you take a general system. So you have your input x. It goes into a box, and z is the output. And you want it to have full change detection. So generally, y is a function of x, y, and z. Dy dt is a function of x, y, z. dz dt is another function of x, y, and z. That's the most general. Dy dt is a function of x, y, and z. dz dt is a function of x, y, and z. So the sufficient conditions are, so what I want is that if I multiply the input by p and the memory by p, I want z to be the same. So in order for that to happen, you want f of px, py, and z to be exactly the same, to be p times f of x, y, and z, and g of px, py, and z to be just g of x, y, and z. That's what I mean by homogeneity. This is the first kind of, it's not exactly, that's like homogeneity of a certain form in mathematics. Now let's just check it, okay? In the case of integral feedback, of linear integral feedback, f is just k z0 minus z. So f of px, py, and z is k z0 minus z. There's no y and no x here. So it doesn't obey condition 1. Condition 1 not obeyed. But let's look at this thing here. Here, f is k, y, z, 0 minus z. So if I multiply x by p, y by p, and z of p, right? F of py, px, and z is p times f of x, y, and z. Because that's why I knew I had to multiply by a y here. It also will work if I multiply an x here. It will also work if I multiply y times exponent x divided. There's a lot of ways to do it. And what about g? If I multiply x by p and y by p, g, this part, remains invariant. It remains the same. So this has full change detection. I know it. The system also has to be stable and have exact adaptation. I didn't say that. And these two conditions. So now if I give you a differential equation model, you can tell me if it has full change detection. It has the sufficient conditions for full change detection. It's easy to check. You look at the functions, you can see. You can generate new full change detection mechanisms. You can be a full change detection expert. Now after this hour and a half, you really know a lot about full change, or you heard a lot. I don't know how much went in. So just any questions right now or thoughts before we finish? Yeah.
So in order for the I1 FFL to demonstrate the MCD,
Yeah.
you need also that the typical time for the signal will be larger than the typical time of it takes your system to reach a steady state. Otherwise,
Okay. So you say for this to have.
overlap.
So you asked about the typical time of the signal. So in fact, the signal can change quickly or slowly. You still have full change detection. And it's not with the steps. With the steps with the step experiment, the way I set it up, it's like you need one step to finish before the other step. But a more general definition of, so this is like one experiment with separate steps. But a more general definition is if I give any input signal, and I get a crazy input signal, I get a crazy output signal. And now I multiply this signal by 2, let's say, I get the same. So even if the signal is changing faster than the response time slower, it doesn't matter. It still has full change detection. That's a more general definition. But in this case, if you give the steps too quickly, then the two shapes won't overlap exactly. I guess that's the root of your question.
Yeah. So we didn't prove it. Just if we use this thing. Then
Yeah, I mean this should be a proof because what it means is that if I double the signal and the system is such that the memory is doubled, then Z stays invariant. That's like saying that multiplying the signal by 2 gives you exactly the same output. Okay, so, but I think for the purpose of this course, if you're not really into math, this part about proving it, it's just extra knowledge that these things exist. What I want you to remember in particular is the incoherent feed-forward loop model. The y dt equals x minus alpha y, and dz dt is x divided by y minus alpha z, right? And let's check the condition, the Chauvel conditions. If I multiply x and y by p, p goes out of before the parentheses, right? So if I multiply x and y by p, f of x, of px, py, and z is p times f of x, y, z in this equation. In this equation, if I multiply p, x, and y by p, they cancel out. So G remains invariant. So this obeys the Chauvel conditions. Yes. It has full change detection. Great. Yeah.
Can you generalize to a circuit with 4 members or n members?
A circuit with more equations. Yeah. So these are actually, in that case, this becomes like vector. Y can be a whole vector. Let's just say 3, 4, 5, 6, 8 memories with one output, and this f is then a vector function. It's the same condition. So I mean, would you like me to write a circuit that has four?
And any
Right, yeah. So it's the same conditions. You can write them down for multidimensional.
If you want 4 letters with arrows between them. Are you familiar with circuits that are big?
No, I never thought to write down circuits with more than X, Y, and Z, like to add a W. So that's could be very interesting. But I think in the world of X, Y, Z, there's this nice work by Chao Tang and Ma that asked numerically, he simulated all possible circuits with 3 components and asked which ones have exact adaptation. So that's of course part of fold change detection. And it turns out that the only two topologies, which are basically incoherent feed-forward loop and a kind of integral feedback loop. So I'm betting you that in the world of 3 components, these are the only 2 classes, I'm not sure, but classes of circuits that have fold change detection. And in the world of 4 components, I don't think I don't know if anything's been done. But it's really, it could be quite, maybe there's additional symmetries, additional, I mean, here speaking again from the physics point of view, in physics a lot of times we're concerned with symmetry. Symmetries can give you a lot of information about a system. Also in biology we're concerned with symmetry. For example, if you know that you're an organism that's moving and what's in front of you is more important than what's in the back of you, you put your sensory systems in the front, let's imagine, okay? But if right and left are symmetric, then you have a right eye and a left eye, and they're not very different. That's like a symmetry, right? So that can determine morphology. And here it's also, there's a symmetry working. The symmetry is that you need to sense something, but if I multiply that something by a number, the output should be the same. So for instance, the visual system, I need to be, the symmetry is a scalar symmetry, is multiplying by the input field by a number. And then you ask, what are all the systems where, which are invariant to that symmetry? Invariant to the symmetry means that if I take right and left and change it to left and right, I get the same, basically the same person almost. Of course, we have the heart on the left. And so invariant to multiplying the input field by scalar is also a kind of symmetry. So this is, you can say, a generalization of the idea of symmetry in biology. And then you can ask questions about other kinds of symmetry, weird symmetries. What kind of symmetries are found in biological systems? And again, that's an open field to explore. Okay, any more questions?
Why is it called integral feedback?
Why is integral feedback called integral feedback? Because, you see, y, the rate of change of y, is equal to the error between z and z0. So in engineering, feedback a lot of times you measure the error, let's say between the desired temperature and the actual temperature, and you do something with it. So if you just take that error, okay, and make the power to the heater proportional to that error, you get what's called proportional feedback. But if you make the power to the air, to the heater change over time is proportional to the error, so that y goes like the integral over time of z0 minus z. I'm integrating this equation. So what y is doing, methylation in the case of chemotaxis, or the concentration of y in the case of incoherent feed-forward loop, is integrating the difference between z and its steady state. Now, why is the integral feedback so powerful? Is because that y, that the memory keeps counting and accumulating as long as the error is non-zero. So as long as the error is non-zero, the integrator keeps counting and pushing the system towards a steady state. So if the error is large, this becomes a huge push, huge push over. Even if the error is small but stays that way for a long time, the integration can be huge. So it's a powerful feedback. That's why it's called integral feedback, because the
formula for the proportional feedback?
It's also integral feedback. So the y is remembering the z0 minus z. But because there's a y here, if I divide by y, dy dt over y, it's actually d log y. So what's going on here is the memory is remembering. How does it go? It's the log. It's something with the log. The memory is remembering the exponent of the error or the log of the error. I don't remember right now. dy dt. So it's log y. Log y goes like, okay. In the proportional, dy dt equals y z0 minus z. If I divide by y here, it's like d log y over dt equals z0 minus z. So log y goes like the integral of the error over time. So, and that's somehow I can't explain it right now, but that's somewhat critical for this fold change detection. Okay, so that's the difference. So biology is easy for biology to build integrators. Basically things like methylation of the receptors, so chemical modification that keeps building up can store information. All integrators, physical integrators, wind up at the end. They fill up the container. So that's where disasters happen, where adaptation stops working, where airplanes crash, called integral wind-up. So you better make your integrator large enough. Yeah, now I understand. This log y can give you many orders of magnitude. Like, so you can, in methylation, for example, you compress the external signal to, the methylation is like the log of the external signal. That gives you, with 5 methylation sites, to go over, let's say, 5 decades of signal. Things like that. Okay, any more questions? All right, this was a very fascinating discussion. Look, the movies are online, so you can see. And let's take a nice deep sigh of relief. And may all your experiences have positive fold change over the next week as we meet next Thursday.